behaviour. Now, it may not be the first thing that comes to mind when you think about microbiology, but bacteria were often, as I hinted at yesterday, the importance of microbes in evolution. Bacteria have often been the first organisms to evolve and develop particular functions. Okay. What's really amazing to me is if you take something like human beings or any real social creature, monkeys, bees, insects, then the different types of social behaviour that they can exhibit are really quite fascinating. Okay? If you share a flat with someone and they don't wash up, what's the reaction to that? You tend to get annoyed, don't you? Somebody's not washing up, somebody's dirty. Some of you might deliberately not wash up because you think somebody else in the group does it. All sorts of ructions take place in second year when you move out of halls and go into accommodation with your best mates, only to find within a few days that they're your worst enemies after you've been living with them for a few days. So social interactions amongst a social creature such as us human beings are very complicated, very important, and they're with us almost every hour, every day. But have you ever stopped to wonder, why is it that we have these reactions? Okay, why is it that we don't like a shirker, somebody that doesn't do their fair share of work in a group? Why do we feel that way? Yeah. Why do we not like cheaters? Why do we not like bullies? Why are these behaviours deemed very negative in cultures all around the world? So what I'm going to talk to you today about is the evolutionary pressures on groups, on social organisms, to behave in certain ways. Okay? So not long ago that human beings were the evolutionary ancestors of human beings were primates that lived in jungles, and you could see that working together socially improves the survival rate and the ability to procreate and have healthy children dramatically. So that if you don't conform, and you don't conform to these social structures and interactions which are designed to help the group rather than the individual, then you would die out. Okay? The grumpy monkey that doesn't do his washing up gets socially outcast by the rest of the group. Ooh, don't talk to him, he's a lazy monkey. And that poor monkey gets outcast, he's on the edge of the group, and he gets picked off by a lion or a cheetah or something. And so Mr. Grumpy Monkey dies and can't have any children. So he can't give birth to lots of grumpy children. So you can see that certain behavioural traits can be removed from the gene pool, and certain behavioural traits can be selected for, so that cooperative behaviour becomes more of a norm. And it's those selective pressures through Darwinian evolution that make us what we are today as human beings and how we behave towards each other. However, as I will go on to show you the origins of those social behaviours and those advantages of working together in a socially conformist way, have their roots, incredibly enough, in very simple organisms such as bacteria. And I think, I might be wrong, it might be worth you looking this up, but I think the only organisms which I'm aware of that don't show any social behaviour are viruses. But I'll have to double check that because it's an interesting question. So I want to talk to you today about the types of social behaviour that can be found in microbes. And I want to speak to you a little bit about the mechanisms of those behaviours. How do they actually work? What makes these behaviours work? And so what? Is that even interesting? Okay, why should we bother to study social behaviour in microbes? Why is that even interesting? Why is that even useful? Is it just knowledge for knowledge's sake, or can that knowledge be applied? And I guess it's a kind of rhetorical question, and you'll know me by now. I like applications of basic science, and of course the answer to that last question is yes, and I'll show you some of those potential applications. So I'm going to make a few film references here, and I'm reminded every year about the growing distance in age between me and the student body. And often say, "Oh, have you seen this film?" And it's quite a recent film, and it turns out it came out like before most of you were born. So just so that I can get a feel for how many of you have seen that movie, just nod or put your hand up if you've watched The Godfather. Quite a low proportion, actually. How many have not seen it? Quite a lot of you. Okay, so background reading. Okay, this is the added reading, which is a lot more fun than going to the library and reading PDFs about science. It's watch the Godfather series. Okay, can you get it on Netflix? Can you watch it free, pirate copy, whatever? Oops, that was on tape. It's a fantastic movie, and for those of you who haven't seen it, it describes the life of an Italian family, which are effectively a mafia family in the US. They come across from Sicily, and the three movies chart the struggles within the family, the bids for power, the murders, and all the social interactions that human beings are capable of. And it really just sums up the complexity of social behaviour in humans. So one of the things that you can see, incredibly enough, is you can see exactly the same types of behaviour being displayed in bacterial cultures or bacterial systems. So bacteria are capable of complex communication with each other, and they are capable, as I'm sure you will recall, of making decisions through a process known as quorum sensing. And although we've treated quorum sensing in previous years and previous lectures, we're going to be talking about quorum sensing at a greater depth in stage 3. So do please revise your earlier activities surrounding quorum sensing, and I'll talk a bit more about quorum sensing today. So did you know that bacteria can be observed to bully each other? Okay, to persecute different populations of a bacterial culture. Did you know that microbes can be observed to cheat the system, and they can take nutrients out of a system without doing any work for the group? And that behaviour has been coined cheating. So microbes, bacteria, even fungi, can display these annoying habits, or annoying or sometimes deadly habits. At the same time, microbes, like more complicated social creatures, can be shown to help each other. They can commit suicide. They can carry out suicidal processes that benefit the greater population. You might have seen this, for example, in ants, when the ants will all link together and make a little bridge across a river. You've seen that on the TV? So ants can make a little bridge across the river. They all tie themselves together, and then the main swarm of ants can walk across the little bridge. So the ants that make the bridge will sometimes die and get washed away in the river. So they're basically sacrificing themselves for the greater good of the colony, which is kind of cool. Human beings do that. They sometimes put themselves in great danger to save others, and sometimes things can go wrong, and people can die when trying to save others. Microbes are no different. Microbes can deliberately kill themselves, lyse themselves with toxins, and then the nutrients that the dead bacteria release can go on to provide nutrients for the next generation of microbes. Aggression, microbes can show aggression, they can detect enemies, they can release attacking compounds and chemicals and antibiotics, and also families of bacteria, families of related organisms will help each other over and above unrelated organisms. So you get little groups of bacteria helping each other if they know they're the same strain or the same culture, or the same family, but they won't do that if they are in a test tube with other strains which they think are a different family. And that family, the families helping each other, was one of the references to the movie The Godfather, which of course is all about particular families helping each other and controlling the supply of money, and the parallel of money, of course, in the microbial world is nutrients. Another question for you. Have any of you read a book called The Selfish Gene? Couple of you. So again, it's by a chap called Richard Dawkins, who in my view is a bit of a wally, but he did write a good book. He wrote a book called The Selfish Gene, and it is definitely worth reading. It describes, in short, it describes the fact that much of biology is designed to protect the genetics of a system. Okay? And if you look at behaviour and the way humans interact and the way plants and bacteria and all these creatures in the world interact, it's about preservation of the genetic makeup of a particular organism. So if you don't have time to read the whole book, at least read the summary of it on Wikipedia, understand about its existence and the general message of the book. If you do want to continue to keep improving your vocabulary and your ability to write eloquently, I suggest that you do go and actually read the book through from cover to cover. Reading isn't just a waste of time, it cultivates improved vocabulary and improved writing skills, which will benefit your exam marks in the long run. So don't neglect to read. A great example of the selfish gene is a phenomenon which I was quite surprised about when I first heard about it, and that is in African lions. If the alpha male of a particular pride of lions dies or is injured or is defeated, and a new alpha male comes into that particular pride of lions, and there are any little lion cubs around, the new alpha male will kill the babies, the baby lion cubs. Have you heard about this phenomenon? A couple of people have. It's quite gruesome. Oh yeah, I'm going to take over this pride of lions. I'm the boss now, and I'm going to look after you all. But I'm just going to kill the babies. I think that's a bit weird. But in fact, it can be understood through the lens of the selfish gene. So because those baby lions aren't the progeny of that alpha male, they don't carry his genes, then he doesn't really want to spend any energy looking after them. And so he kills them. And then the new babies that are born are his genes, his genetics, and then he selfishly promotes his own progeny. So that's quite a sort of violent illustration of the selfish gene. But the more you look at that topic, the more you can see examples of the DNA and the genes of a particular system being preserved through various actions of biological systems, which is really quite fascinating. So there's just a sort of rather frivolous cartoon about bacteria forming groups and behaving negatively towards each other. And as I've said, social behaviours are not unique to complicated organisms. And I've said here they can be observed in bacteria and even viruses, and I'm querying the viruses one because although I wrote that here, I've not been able to find concrete evidence of viruses showing complex behaviour. The key point here is that if you work together in a group, you can often have a significant advantage over working together as an individual. Okay? And a really simple example of that would be catching your food. Yeah? If you are a monkey, again, in the jungle, and you're trying to catch a bit of food, and you're a big monkey, and you want to eat a little monkey, and you try and catch a little monkey to eat, then it's going to be quite tricky, because that little monkey's going to run away. If there's 6 or 7 of you, yes, chasing down that little monkey to eat it, then you've got a much higher chance of success. You can gain food and nutrients much more readily if you're working together as a team. Okay, so teamwork provides you with a really important advantage over an individual. Okay. So the social network then is a process which can encourage or enhance group behaviour. Okay? And that's really important to bear in mind. Now, here's a fantastic example of group behaviour in bacteria. And I'm describing on this slide, of course, the biofilm. So these are individual cells which are swimming around on their own, and they're not gaining any advantage by swimming around on their own. They're sat there. It's just them against the world. Okay? What can happen though is that then when they become attached to a surface, they will differentiate and they'll start producing matrix polymers, polysaccharides, proteins, extracellular DNA. They will bind together and start working as a group, as a social group of organisms. Okay? That will grow, and then the biofilm can continue to function. But what are the advantages of doing that? Well, first of all, they can protect themselves by this big layer of slime, where they're all contributing to a protective layer. All these cells are providing a home for the main population, and that slime can attract nutrients, and therefore the nutrient levels in the biofilm tend to be higher than swimming around on their own. And also the environment within the biofilm can change. So the pH of the biofilm can change to a more suitable pH. And the chemical complexity of the matrix can also provide some degree of chemical buffering against changes in the main water body. So if there are changes in the pH in the main water body, then those changes will be buffered against inside the biofilm. So what we're seeing here is we're seeing the ability of bacteria to behave in a large, complicated group of organisms, which provides them with the ability to control their own environment and protect themselves much more readily than if they were just swimming around as a single cell, all on their own, in the middle of the ocean or in the middle of a lake. Okay? And of course, they can also extricate themselves from that situation. Okay? They can, if the nutrient levels fall and they think, well, this is not a nice place to be, they can release enzymes or D-amino acids or other types of agent which will serve to disperse that biofilm. Okay? And it's the same with us. If we can find ourselves in a really cool social group, you might want to form a team to have a football match, for example, and you're going to win that football match because there's 11 of you rather than one of you. You might want to form a study group and all go to the library in a little group of 4 or 5 students, and then you help each other. You form a small social group that can help each other to revise, and that's a really great thing that I would encourage you to do. But you can also extricate yourself from a social situation. You can stand back, you can go away, you can go to the library, lock yourself in a room on your own. So you can do both. And that's exactly the case also with microbes. They can form these complex social groups, or they can behave in such and such a way whereas they can extricate themselves from these social groups and return to their solitary lifestyle, in this case swimming around on their own. So the parallels are really quite fascinating. In fact, if you do look into human psychology, what's really interesting is that human beings will often behave very differently when they are acting as an individual compared with when they're acting as a social group. And some of you might have seen these studies where they've taken film of individuals in a room where the person is on their own. Yeah, do they steal sweets from a table? Do they not steal sweets from a table? And when they're on their own, they will often do more selfish acts. Okay, a single human might steal a bicycle, for example. Human behaviour changes significantly when that individual human understands or recognises that they are being watched by other human beings. Right? So if you're in a room and there's 5 other people, you are much less likely to steal some sweets from a table. Interestingly, oh no, don't want to show myself up as a thief, as a baddie. But if no one around, yeah, fair game, I'll have some sweets, right? So your behaviour changes depending on whether you think you're in a social group or not. And when you think you're in a social group, you behave a lot more socially. And that's why, I don't know if you've noticed around the campus, a lot of the bike sheds have got a big pair of eyes printed out and stuck above the bike shed. Have you seen that? And that's because some really interesting work was done where they noticed a dramatic fall in bike thefts when they put this big pair of eyes above all the bikes, because of this subliminal feeling that the thief felt that they were being watched by another human being. And so they were more reluctant to steal a bike when they had this big pair of eyes watching them, which I think is fascinating. And again, it describes how social pressures, even subliminal social pressures, can change dramatically our own behaviour, and that also happens at a microbial level. And you remember when I mentioned yesterday, how long have microbes been on the planet Earth? Just yesterday. 3.2-ish, yeah, about 3 billion years. So many billions of years. So they've had some time to hone and polish and improve these processes, and I'll go on to talk to them out. So biofilms, okay, just underline that. Biofilms are a fantastic example of the evolution of social behaviour in prokaryotic bacteria, and I've given you some examples of how growing as a biofilm can be evolutionarily beneficial. And again, here's a very nice article from Trends in Ecology and Evolution. And this is from way back in 2001. So you can see that this has been studied for a number of years. This is about the evolution of social behaviour in microorganisms by Crespi. And again, these are further examples of social behaviour. And this is a picture of a fruiting body of a Myxobacteria species, and these organisms can change their behaviour of certain groups so that cells can fruit and sporulate and improve the dispersal of that species. Okay, so some cells are acting as a dispersal body, okay, and you can see now the evolution of dispersal, the evolution of fruits and seeds. Okay, that already started in microbes, and that required specialization. It required certain groups of cells to behave in a different way, to communicate in a different way, and to develop in a different way. So that's a really nice example, dispersal. Now the other one is predatory mechanisms, which is a really nice one as well. Okay, This is about bacteria eating each other. And of course, that's a very strong selective pressure. If you get eaten, you can't pass on your genes to anything or anybody. And here are just 4 examples of predatory behavior in microbes. So the first one is phagocytosis, of course, which I think you should already know about. If you don't, just do some quick revision after the lecture, in which a purple eukaryotic cell will engulf a bacterium shown in yellow, and it will be absorbed into a vacuole, and then the eukaryotic cell will release degradative enzymes into the vacuole, and that bacteria will dissolve, be broken up, and all the nutrients from that bacterium will be absorbed into the eukaryotic cell. So that's a very simple evolutionary progenitor of the stomach, isn't it? Yeah, you eat the foods, you digest it, and you absorb all the nutrients. Now, another one in B is a really cool one. This is the attack. The yellow cell is being attacked here by the little white cells, and the little white cells are called Delovibrio, with the silent B on the front, Delovibrio bacteriovorus. Okay, you may have heard of the Latin word voracious appetite, where you eat voraciously, so that means eating bacteria. And so that Delovibrio will start to degrade the yellow food cell, and it will absorb all the nutrients, and then lots of little Delovibrios will grow out of that organism. So this is a bacterium that eats other bacteria. In C, we've got Streptomyces, which is a type of filamentous bacterium, producing antimicrobial compounds which leach out and kill and lyse other types of bacteria. And of course, once the other bacteria have been killed and lysed and are dead, the nutrients released from that attack can be absorbed by the Streptomyces. And then finally in D, we've got predation. This is a bit more aggressive. This is a strain of Myxococcus, which are the orange cells, and they will literally invade the other colonies. They'll burrow in and they'll lyse it, release hydrolytic enzymes to destroy and kill the bacteria right next to them, rather than using a kind of long-range secretion of antimicrobial compounds. So they are, 4 concrete examples of predation and how social behavior in bacteria can lead to the absorption of nutrients through predation. And again, bear in mind these are just some concrete examples, of course, that I'm delivering to you in the lecture. I think what would be great, what I would expect of a student which would deliver a first-class mark at the end of the year, would be for you to go away and perhaps pull out a couple of other examples from the literature, go into a little bit more depth of some of these examples, so that when I'm reading your answers in the exam, I can see evidence that you've gone away and done that extra reading. Okay? A common problem in student work in exams is that students may do some further reading, and they may print off loads and loads of papers and keep them in their bag for months and cover them in yellow marker pen and learn all about the stuff that's in them and do really great reading, but they fall down at the last hurdle in the exam room when they're writing about a particular topic. Okay? An exam question may well be discuss the benefits of social behavior in microbes. Okay? And then as an examiner, I cannot see any evidence on the paper of that additional reading. So if you do additional reading, practice incorporating it into your writing. Okay? And again, that might be something you could do in small study groups. Yeah? Where you do that, you do write little half essays or quarter essays, and you show them to each other And you say, well, where is evidence of additional reading? Okay. And again, I'd point you to the faculty marking criteria. Have you seen that? Only very few nods in the audience. Okay. I would strongly recommend that you get hold of the faculty marking criteria this week. Print it out and put it very prominently in your notebook or on your fridge or above your headboard or somewhere very important, because those marking criteria are your guidelines of how to do well in exams. And believe you me, if I'd had those marking criteria when I was at university, I would have loved that and I would have been all over it very early on. Okay, and I'm staggered actually by the number of students that don't read them and have just sort of kind of vague recollection that they might be on Blackboard somewhere. Get hold of them, read them, use them to self-mark your own work, and question your work and what you're doing. Critical reading, critical analysis. Yeah, have you done some of that? Have you incorporated that into your work? And that critical analysis starts here. It starts now in this lecture when you start critically thinking about what I'm teaching you. Okay? And let it flow on to when you're reading papers, critical analysis of those papers as you read them and as you take notes on them. So there's just a few little top tips there. So this is just a further example of seeing these processes that I showed you in that slide, looking at them under the microscope. So again, A is a time course predation by these cells. You can see these cells come in contact to these cells, and you can start to see the breakup. That cell, which is nice and whole and clear in the first photograph. This is a time-lapse photograph. If you look to that one, then to that one, then to that one, you can see that cell has completely exploded and blitzed. Okay. Equally, in B, we've got a time course of M. xanthus invading and lysing a colony. So that's M. xanthus. They're moving across there. And you can see that the clear part is where they have broken open all of the cells. Okay? And you can see that again, with time lapse, that's amazing. It's like a sort of a wave of attack as it goes across and kills all these organisms. And then finally in C, we can see that there are three types of group-mediated predation, and then the rippling effect of this one here as well, forming the fruiting bodies at the bottom, okay, which is one of the ones that I've told you about. So these can be observed under the microscope, and in my view, they're equally as dramatic as large animals killing each other on the African plains. These are very sinister types of behaviour where one population of microbes can overtake and kill another population of microbes. So this next table is again really quite interesting because it talks about the topic of convergent social phenomena. Okay. So that means different things can evolve on separate occasions for the same kind of purpose. And that's called convergent evolution. So a good example is domicile creation, making a home. Okay? And I've told you that in microorganisms, they make homes by forming a biofilm. If you look in animals, well, what do they do? You could probably write me thousands of words on how animals create homes. They produce burrows, nests, hives, galls. There's lots of ways in which animals can produce homes. Cooperative hunting. Okay? So I've told you myxobacteria and some pathogens can hunt and kill other bacteria. We know about cooperative hunting. Wolves, lions, beetles. And there's some fantastic footage, I don't know if you've seen it, I think it's one of the David Attenborough Nature series of African hunting dogs. And it's aerial photography from a helicopter, I think, which is extremely high up in the air. And it's so high up so as not to cause any disturbance to the African hunting dogs. And it has a kind of gyroscopically stabilized camera looking down on these hunting dogs. And you can actually see them fanning out around some prey, and they kind of communicate in some mysterious way. Or you go that way, I'll go this way, you go straight on, and we'll all converge and we'll kill the prey, you know, a couple of 100 meters away in the bushes. And it's spectacular how smooth and well coordinated that behavior of attack is. And it's really interesting to understand that that's clearly evolved right the way back through simple organisms, through beetles, all the way back to bacteria. Some organisms can be specialized providers of food. We spoke about that yesterday. We spoke about rhizobium producing fixed ammonia for plants. Okay, so that's a type of social behavior or social interaction. So I think, again, it's important to look across these lectures to find themes and not to think of lectures as bodies of knowledge in little boxes. Okay? These themes often cut across different types of topic. There's specialized dispersal, there's altruistic suicide, something that I've mentioned. This also occurs, for example, in honeybees, wasps, and pea aphids. And also, of course, communication via chemicals, quorum sensing in many bacteria. What is chemical communication in higher organisms? So again, hopefully all of you have heard of things called pheromones. Okay, so moths can communicate with each other through pheromones. Humans communicate with each other through pheromones and smell. It's a very important aspect of communication. Trees can even communicate with each other through releasing chemicals. When trees are under attack from beetles or aphids or even disease, often those trees will emit a kind of "I'm being eaten by beetles" chemical signal, and that signal goes into the air, and incredibly other trees can absorb that signal. They can detect those molecules, and they can increase their production of anti-insect chemicals, for example, in the tree. So it's a chemical defense, which I don't know if you knew about that, but when I first heard about that, I was like, wow, that's incredible. Trees can communicate, and they can help each other by sending out these important, quite complicated messages that change the behavior of the trees, you know, further down through woodlands. So, incredible topics with their roots in microbiology. And as I've said, I do want you to revise quorum sensing. I do want you to know about quorum sensing in detail. The more detail, the merrier. These slides just serve to briefly remind you that quorum sensing is a form of communication in both gram-positive and gram-negative bacteria. The molecules of communication are different depending on whether it's a gram-negative or a gram-positive bacteria. They are small natural products such as AHLs in gram-negative, whereas they're peptides in gram-positive. Anyone remember what AHL stands for? Ooh, that's year 2. That's like before the summer. Anyone? What does AHL stand for? I should bring in some prizes to encourage you to shout out the answers. No? A little bit far away in memory. Acylated homoserine lactones. Okay, so that might ring a bell from earlier years. And the fascinating thing about quorum sensing is it provides a so-called density-dependent trigger. And that means that this serves to allow the bacteria to take important decisions. Okay? Are we going to spend lots of energy on infecting that person? Are we going to release lots of virulence factors? Are we going to release lots of enzymes and cause an infection? Because that's quite hard work, to be honest. And if we're going to fail, and we're not going to succeed in causing that infection, we might as well not bother. So that's that's what's happening in the bacterial population. If there's a higher number of bacteria, then the probability of successful infection is greater, and therefore a decision point is reached where the bacteria say to themselves, well, if there's lots of us, we'll go for it. We'll cause an infection. Okay? You can see that very clearly in other bacterial behaviours such as luminescence. If there's not very many cells, they're not going to bother producing luminescent light. If there's lots of them in a high density where the luminescent light production will have an actual effect, then they will go ahead and produce light if the density is high enough. Okay. There was just driving in this morning, there was an article on the radio about jackdaws. Again, this is probably in the press if you want to look out for it. So jackdaws, they protect their nests by mobbing other birds like magpies or sparrowhawks. They mob them and chase them away from the nest, which is a selective process which allows a group of jackdaws greater abilities than a single jackdaw. So they mob the attacking bird. And what this scientist Laura Coombs showed from the University of Exeter is if there's lots of jackdaws, if there are more than 5 jackdaws, then other jackdaws will come along and join the fray and bundle in and try and chase off the baddie. But if there's only 1 or 2 jackdaws, then they won't do that. Yeah. So again, decisions taken on the basis of safety in numbers, but again carefully communicated by vocal calls. So this idea of working together for the benefit of the group runs right through the whole of biology, and of course it began in these organisms. So again, I won't belabor the quorum sensing. Suffice it to say that you should be aware of and be able to explain the different types of behavior, low density and high density in the case of both gram-negative and gram-positive. The fact that these cell densities can have a triggered response which leads to the transcription or the non-transcription of certain target genes, including, for example, virulence enzymes or fluorescence proteins, for example. So do look into that and make sure that you know that and can provide carefully argued scientific reasoning surrounding the importance of quorum sensing in communication and in social networks. So again, I think this question has come up in previous years in exams, and again, when students are responding to an exam question on this, some of them will even, they'll forget to mention really major issues such as quorum sensing. Okay, and I'll be looking for that in an exam. Quorum sensing is a major communication mechanism which allows social behavior to take place in microbes, and a good example of that is a biofilm. So, quorum sensing and its role in biofilm development is a great topic. I would suggest for a little bit of further reading. Now, here's a really nice slide which I always like to show, because this shows a picture of my colleague, a Dutch scientist called Reindert Nijland, Nijland, and he was successful in securing a 2-year EU-funded postdoctoral fellowship to come and work with me at the Dove Marine Lab a number of years ago, and we discovered that bacteria can communicate through airborne signals, which is basically the fact that bacteria can smell each other, and that wasn't known prior to our work. So we reported the discovery of olfaction in bacteria, a sense of smell, and again it stands to reason that if you go back through evolution, it would have been very, very useful to be able to detect other living organisms, okay, in order to avoid them or in order to attack them, or in order to gain benefit from that knowledge. So we showed that ammonia signaling, so bacteria can smell each other, and when they smell each other, they're using the molecule ammonia. And so some bacteria can detect ammonia production by other bacteria, and then that triggers a behavioral response again. Okay? When you walk into a room and the room is empty, you will behave in a certain way. If you come into this room this morning, you look around, the room's empty, you will behave in a certain manner. If you walk into this room and there are 10 angry people with knives running at you, you're going to go, ooh, this is a bit weird, it's a bit different, and you'll probably run out the door the opposite direction, right? So your behaviour will change quite dramatically depending on what you detect. If those 10 people with knives weren't running at you, but they were sitting there extremely quietly, and you were blind, you wouldn't know that they were there, so you wouldn't change your behaviour. So what I'm trying to get at is that the detection, the ability to detect other living organisms has a great impact on whether you might be able to survive a situation or not. And therefore you can see that the ability to detect other organisms would have evolved very, very early on. And we actually discovered that with some simple experiments down at the Dove, which we were very excited about, and got a lot of press and media attention right around the world. So we were really pleased as well, because we weren't the only group to do this. About a year after we published this work that bacteria can detect each other through the release of ammonia, a very prestigious research group at the Institut Pasteur in Paris also showed that ammonia can be detected by bacteria through the air at a distance through a process which we've called olfaction. And that can actually lead bacteria to become more resistant to antibiotics. So I told you the story about the trees releasing chemicals, and then they upregulate their production of anti-insect compounds. Here is it happening in bacteria. Bacteria can detect the presence of other bacteria. Effectively, they get a bit scared, and they go, ooh, there's other bacteria over there. I better try and defend myself. And so that process upregulates the production of antibiotic resistance, yeah, which is incredible. The bacteria can smell each other, and when they do smell each other, they increase their ability to resist antibiotics. That's just an ecological phenomenon. Think about what might be happening in a hospital. Ooh, yeah, hospitals can get a bit muggy. We keep all the windows closed, don't we, in the central heating up. And the air doesn't get changed much. So if you go into a hospital, it kind of stinks a bit. And what you're smelling is the presence of other human beings. Okay, we are exuding carbon dioxide, we're exuding ammonia, we're exuding lots of chemical signals that indicate the presence of a living organism, and bacteria are in hospitals. So when they detect any ammonia in the atmosphere whatsoever, what's going to happen? They're going to become more resistant to any antibiotics that are used. So this knowledge has important implications for understanding and treating antibiotic-resistant bacteria in a hospital environment. What did the Victorians do prior to the discovery of antibiotics? They had hospitals where the nurses would come in every morning, throw the doors and windows open, and let some freezing cold air in. Yeah? And through trial and error, they knew that fresh air was beneficial, and there was less sickness in the wards when there was more fresh air. Are those two things connected? Probably. So what I'm excited about is that basic study of bacterial physiology can have really important implications for the way that we treat patients and fight things like antibiotic resistance in hospitals And you know, these things are really deadly. They're getting worse every year. We're kind of getting numb to the fact that people go to hospital these days and die through infection or sepsis, particularly in the elderly or the immunocompromised or the very young. It's happening much, much more often than it did, say, 10 or 20 years ago because of the rise of antibiotic resistance. And maybe things like this are relevant to that. So there's the paper, Bacterial Olfaction, published in 2010. And this is a photographic illustration of that. This is a microtiter plate, which is a plastic dish filled with lots and lots of little test tubes. Here we've got bacteria growing in a relatively high nutrient condition. And here we've got bacteria growing in a relatively low nutrient condition. So these bacteria are growing up on the nutrients and they're producing ammonia as a waste product. That ammonia is rising up into the air, it's diffusing across, and the bacteria that are closest to these bacteria are detecting that ammonia. And they're responding, as you can see, by releasing red pigment. And I think you can see in some of the boxes, there's also some very clear formation of gooey biofilm. So these bacteria are detecting ammonia. That's triggering a defense response in them. They are protecting themselves by clumping together immediately into a biofilm. And they are doing another thing. They're releasing this red antimicrobial compound. Okay, so this is a very interesting and complex response. They're protecting themselves to defend themselves, but they're also releasing antimicrobials to attack any potential predator or perceived predator. Okay? And that photograph, where some cells are sort of slightly pink, is really a fascinating photograph which illustrates complicated social defense and attack responses in bacteria. And you know, you can get additional information from the paper which looks much more carefully at the detection of ammonia in these organisms. This was just some of the press that was associated with that. If you look more closely, this was the work done by the French group, Jean-Marc Gigo's group in Paris, the upregulation of antibiotic resistance. And I won't go into the details of that paper. I will allow you to read the fascinating experiments that were done by Gigo's group during that work. And again, if you look more closely, how is it that bacteria can detect ammonia? Right? How do they detect ammonia? Well, they detect ammonia through, funnily enough, proteins on their surfaces. Okay? This protein here, called AMTB, is a receptor protein which exists on the surface of the bacterial cells. And that protein will bind ammonia, and then there will be a little biochemical cascade which triggers a response. So that's actually the bacterial nose. That is the organ, the protein, whereby the bacteria can smell or detect the presence of ammonia. And as a result of that, we have a glutamate dehydrogenase, glutamine cycle, and the production of a biochemical cascade. So as soon as ammonia binds, a biochemical cascade is triggered, and a behavioural response results. In exactly the same way, again, if you're walking down the street and you smell a pizza and you're hungry, you'll be like, yes, I think I'll walk in the opposite direction and go towards that pizza shop and get a pizza, because I'm hungry. So smell, detection of certain molecules, changes your behaviour very dramatically. Ooh, that smells awful over there. I'm going to go in the opposite direction. Yeah? Bacteria were doing that 3 billion years ago. So, again, to finish off, our knowledge of these mechanisms can help. We know about quorum sensing. What can we therefore do to stop bacteria causing infection? We can stop quorum sensing working. We can use so-called quorum quenching enzymes. So this lactonase breaks down AHLs, stops the bacteria communicating. So you can jam communication. You can stop bacteria talking to each other. When governments get annoyed with people, human beings, causing riots, then one of the most common modern tactics is to switch off the social networks, switch off the mobile phone network so that human beings can't communicate with each other and coordinate their dastardly deeds.